Trojans Talk Research is a monthly spotlight on the research interests of Troy's faculty, presented by Troy University's Office of Sponsored Programs. This month, Dr. Jacqueline Jones presents her research on the effect of the tumor microenvironment in promoting prostate cancer bone metastasis.
So I think that y'all know Dr. Jones. She is going to be the, have you heard the news?
What?
You're the new chair.
Oh.
She's going to be the new chair of bio starting in July, and right now she's coming to you in one of her other roles. She's one of our great researchers. She has a very active research program where she has been doing cell culture here for the past 10 years, 12 years?
Not yet, 8, 9.
All right. A while. She's been maintaining active cell culture here, so we're very excited for her to present her research.
Thank you.
Good luck.
All right, so as Dr. Hardin said, I work with cancer research. Today I'm only going to present on prostate cancer and our role in looking at FOXC3, which is a known bone regulator to promote prostate cancer. So what is prostate cancer? Prostate cancer is the abnormal growth of cells in the prostate gland, and it is the second leading cause of death in men under the category of cancer. The vast amount of research that has been conducted in the field has led to curable advances and treatable advances if detection occurs early. The issue is with late stage prostate cancer, which sometimes can be asymptomatic, the more aggressive it becomes, the reduction in survival rate. And so what we've identified specifically and what my lab focuses on is the advanced stages, because early detection, there's a cure, there's treatment out there. But when it becomes aggressive and it metastasizes, I'm gonna go between researcher and teacher, okay? When it moves or metastasizes to another location in the body, that's where it becomes difficult and the mortality and morbidity rates increase. One of the places that the prostate cancer prefers to metastasize to, you have the liver, the lungs, and the bone. If it goes to the bone, death is inevitable, okay? So at that point, if someone has metastasis to the bone, the goal is to increase their lifespan, whether it's 2 years, 3 years. So in order for a prostate cell to become cancerous, it has to change all of these genetic, molecular, morphological changes to become aggressive. I want you to notice the left side, yes, is showing an epithelial cell, okay? Those are your nice, normal cells. They have structure, they're organized, they show cell polarity, meaning they have an apical and basal layer. They're excreting and showing what we would call cell adhesion markers that allow them to stay uniform in a controlled environment. In order for a cell to become cancerous, it has to go through a transition that we call epithelial to mesenchymal or mesenchymal transition. And what that transition means is it goes from a normal-like cell to a unorganized cell, almost like stem cell-like. Very plastic. You'll notice on the right side that it is elongated. It no longer has structure, organization, and a lot of those cell adhesion markers that I mentioned are decreased. And when it goes through this transition, the cell is able to move. So what you see here on this slide, at the top, you see that there is a tumor. There's normal tissue. It goes through that EMT transition, and now it's able to invade the basement membrane. The basement membrane, as you'll see in the third portion of the slide, allows the cell to intravasate into the vasculature and use the vasculature to transport itself to another place in the body. So once it gets to a secondary site, it extravasates, it exits the vasculature, and establishes a secondary niche or tumor. And so what our role is we want to identify and understand this complex and dynamic process. So as I told you earlier, prostate cancer's preferred site is the bone. and I want to help you guys appreciate bone a little bit more and understand why prostate cancer prefers to go to the bone. When this prostate cell leaves the prostate gland and metastasizes to the bone, it's one of the most rich regenerative organs in the body. It's composed of myeloid cells, bone marrow cells that are heavy in secreting growth factors, and we like to say that it's a good soil for that seed, that cancer seed, to grow. And so as the cancer cell establishes a secondary tumor in the bone, it's believed that it engages in what we call osteomimicry. Fancy way of saying it mimics bone cells. The reason why it chooses or stimulates to mimic bone cells is because it allows it to evade the immune system. And so as it's evading the immune system, the tumor can silently grow. And this is what leads to the high mortality and morbidity rates I was explaining about. So one of the things that I looked at first in a collaborative relationship with the University of Michigan is we looked at myeloid cells, monocytes and macrophages, because it's known that these cells are capable of helping tumors to grow. Another term for it is TAMs, tumor-associated macrophages. And so they're categorized into two types, M1 and M2. So what I wanted to do was first identify what happens if I give mice tumor, prostate cancer, and then I give it a drug that specifically targets macrophages and monocytes. What will happen? So I inoculated the mice either with the intracardial injection of prostate cells or intratibial, directly into the bone, allowed the tumor to establish, and then gave it treatments of a drug called trabectedin. And over 6 weeks, I monitored the tumor burden of the mice over 6 weeks. And you'll see when you compare the left side, day 0 to day 42, what you're looking for is that the bluish coloring. You see that it's significantly decreased. So the question I had was not so much what is trabectedin doing, because we know what trabectedin does. My question is, what is allowing for these macrophages to stimulate the growth, and what molecular mechanisms are occurring there? So we then, I obtained blood samples from men that had advanced stages of prostate cancer and compared it to men that did not have any cancer at all. And I looked for the monocytes population in their blood, because we want to identify, is there a unique population of monocytes that may be an indicator for prostate cancer bone metastasis or advanced metastasis. And we were able to identify that there is a unique population of what we call PBMCs in the blood that are directly associated with advanced prostate cancer. So you may be asking, as T. Roy is showing you, what is regulating the production and the secretion of these factors that promote prostate cancer? Because we know it's well established, if tumors are there, there's macrophages, and they're saying grow, grow, grow. But what is directing this behavior? So this brought me to FOXC3. FOXC3 is a fork-head family transcription factor that has the ability to bind to DNA in two ways, either the major groove or the minor groove, and regulate gene expression. So we wanted to look at this protein specifically because it has not been studied at all in cancer at all. And we know that it plays a significant role in wound healing, embryogenesis, development, and bone regulation. Specifically, I just gave 2 examples. There was a study with canines, there's been studies with zebrafish, and with humans. When FOXC3 is abnormal, it causes a retard or an abnormal bone stage of bone development. And so, given all of these factors, I said, there has to be some role for this protein in promoting cancer, specifically prostate cancer. So, what's the connection between this protein I just told you about, FOXC3, the bone environment, and cancer? Remember this slide? When tumors go to the bone, it's not just bone that they see. There's this heterogeneous population of cells that are able to break down bone, resorb bone, build bone, And then there is macrophages present, myeloid cells. And I want you to think about this. This is not an abnormal process. None of these things are. They happen, we need them. It's just that cancer cells are so smart that they hijack normal processes that we need to substantiate their growth. And so the bone is a prime soil for prostate cancer to grow and to thrive and to go undetected. And ultimately, as these processes that are normally happening in the body occur, it creates a vicious cycle that just keeps feeding the tumor. So, some initial studies by some former graduate students was to look at FOXP3 expression in cancer. It didn't matter if it was lung, bone, prostate, breast. We just wanted to identify what is its expression levels, because at this point no one had studied FOXP3 in prostate in any cancer. So we did a secondary analysis pulling data from CBio portal on clinical data, and we looked at various cancer types to see FOXP3 expression. And what we found, outside of, I believe it was thyroid and colorectal cancer, FOXP3 expression levels were elevated. We then specifically targeted cancers that had metastasis to the bone, and it was only when we looked at the bone environment that we saw elevated levels of FOXP3. So we then did SAM, which is a significant analysis of microarrays, and we took two different studies, and we looked at FOXP3 expression in normal, which was considered wild type on the left side, and on the right side it is knockout. So the FOXP3 was removed. Red or warm colors indicate high levels of expression. Blue, cool colors indicate low levels. And what we see, FOXP3 is high in the wild type, but as it is knocked out in the various model systems, its expression is decreased. What's unique about this analysis is those genes, there's about 100 or so genes there. A lot of the genes that showed a significant reduction were associated with promotion of advanced cancer. So now we have to take that secondary analysis, that bioinformatics, and we have to apply it to verify if what we're seeing is true. So we used 3 different prostate cancer cell lines, 2 of them from human males, 2 of them are bone metastatic cells, human cells, and then 1 is metastasis to the brain. And you can see from the picture there, as the cancer cell becomes more invasive, there's an increase of expression of FOXP3. So we next wanted to identify, is there a clinical relevance of this in humans? So we did immunohistochemistry. That is a technique that allows you to look at protein expression in human tissue. We obtained about 240 samples of human tissue varying from normal all the way up to metastatic. And we used a staining. The staining for you is you want to pay attention to the red staining, alkaline phosphatase. And you'll see that as the cancer becomes more aggressive, going from left to right, top to bottom, the expression of FOXP3 increases. So as the tumors become more aggressive and advanced, the expression level of FOXP3 increases. We then did an analysis where we quantified the expression level of the expression of FOXP3 in each tissue type, and then we did a quantitative analysis to see if there was a correlation. And we looked for clinical patho features that would be relevant to what we were interested in. And grade, Gleason grade, and even the presence of whether it was neoplasic or not showed an elevated level of expression of FOXP3. So this was promising. So to go and double back on this, and I want to explain why we did another analysis using TCGA, which is the Cancer Genome Atlas program. It's a way to understand the molecular mechanisms in the cancer genome. One of the issues that plagues scientists that study prostate cancer is men don't go to the doctors enough. Sorry, guys. And it's not easy to get tissue. So oftentimes when there is clinical trials or studies that are done, it is required to be available to the public for scientists to do secondary analysis on. So you'll see here, we looked at different studies And we wanted to identify FOXY3 expression in bone metastasis. And then we wanted to identify if there's a correlation with one of those growth factors that we saw prominent in the bone with this protein. So we did several analyses, and ultimately what these diagrams show is as the cancer becomes more invasive or aggressive, there's an increase in FOXY3, but it's also correlated with an increase in FGF8. FGF8 is one of those growth factors in the bone that says grow, grow, grow. So now we have to go back to the bench to confirm what we saw computationally. And so we took those 3 cell lines and we treated them with FGF8. We then isolated, we first did a expression level. Let me make sure I'm seeing this right. Yes, we isolated the RNA from the cells, pre and post treatment. And then we did real-time PCR and we did Western blot. And you can see here, the first few show that even when we add or treat these cells with FGF, there's an increase in FOXY3 expression. However, we don't see that much of an increase in the DU145 cells. Those are the brain metastasis cells. So this is even intriguing because what this says is this behavior is most likely attributed to the bone. So what do we have to do? We want to look at the functional aspect of FOXY3. Right now, all we've shown you is levels of protein, level of mRNA. But now we have to look at the function of these cells, right? Because these cells have to be able to functionally move or metastasize. So 3 of the indications of invasiveness is the ability of the cells to proliferate, meaning to grow, double. The ability of the cells to migrate, that means move, okay, from one area to the next. And then the ability of the cells to invade. Now, I want to explain why this is important and how this ties back to gene expression. All of these functional behavior that I just talked about still tie back to the genes. It's regulated or stimulated by a protein that has to be turned on or off. So the top 2 graphs you see is where my students, prior students, have treated the cells with FGF8, and they looked at the ability of these cells to proliferate. And interestingly, FGF8 causes the increase in proliferation. The next set of, I think it's C, we did a wound healing assay. What a wound healing assay is you plate the cells about 80% confluent on the plate, and then you create a wound on the plate, which is a denuded area, and then you wait for the cells to move or migrate into that wounded area. And we measure quantitatively that denuded area over time, okay? So when we treated the cells with FGF8 and then measured that wound healing, the ability to migrate, we see that FGF8 treatment, growth factor, stimulates the cells to migrate or to move. And it's indicated by the denuded area closing in, okay, and getting smaller. And so what we then needed to identify, as T. Roy asked, what if we decrease FOXY3? We see that FGF8 is responsible for stimulating these aggressive behaviors, but what happens if we remove FOXY3 out of the picture? Will we see a change? So we did a transient knockdown first. A transient knockdown is where we use siRNA to knock down or decrease the levels of FOXY3 in the cell. And what this siRNA does is it degrades the mRNA so that the protein cannot be expressed. The issue is, and why we say transient, is because it only lasts for about 8 days. Okay, so it's not stable. All right, so go back to that previous slide where I showed you all those functional behaviors, right? We said it has to proliferate, meaning grow, it has to be able to move or migrate, and it has to be able to invade. So we removed FOXY3 from the equation. We knocked it out, and then we treated it with or without FGF8 to see, do we see the same behavior? And what we found is that even when we treat the cells with FGF8, when FOXY3 is removed from the picture, it is not enough to recover that behavior. So this suggests that FOXY3 does play an integral role in its ability to stimulate invasion, metastasis, and more aggressive forms or behavior of these cells. So what about the bone environment? We've been talking about the bone, right? Where does the bone fit in, and how do we look at the bone? Remember the study that I showed you earlier with Michigan, where I inoculated the mice with tumors, and then we treated them to get rid of the macrophages. We took that same study and we used the tissue from the mice to look at FOXY3 expression. So the first thing we had to do was establish a baseline. What does FOXY3 expression look like when there are no tumors, just treatment or no treatment? So this is again IHC, and what you're looking for is the brown staining. The brown staining is the chromogen for the expression of DAB. And you can see in vehicle is non-treatment, so the mice were given saline and then treated. They're given trabectedin, that chemotherapy drug. We see a increase, or we see expression of FOXY3, but statistically it was not relevant, okay, which we expected. FOXY3 is present in the bone. So then I took the two different mouse models, the tissue from the mouse models. The first one I used was the intratibial injection. So ultimately what happened, we took the mice, injected into their intratibial right here, the cancer cells, and then we treated with the drug. We then used the tissue to analyze FOXY3 expression. And you can see the top, again, the vehicle treated is saline, the bottom is trabectedin. And the relevance of this is if we remove the macrophages from the equation in myeloid cells, what happens? When we do that, we see a decrease in FOXY3 expression. Again, this was quantified through a technique that we use and it's well established to identify expression levels, in addition to a pathologist scoring the tissue. And it was significant. So that was what we call an ectopic transplant. So I just took the cells and put them directly into the tibia and looked at expression. The intracardial injections were where we injected it into the heart of the mice and then waited for them to show normal signs of metastasis. It would mimic metastasis in the body of a man, okay, male. And so what we see here is when we look for FOXY3 expression, unfortunately, we did not see a significance. One of the reasons why I don't think we saw significance is, one, we may not have caught the tissue at the right time, because it's a highly complex process. And remember, it can go asymptomatic or dormant for a while, and so it can actually modulate the expression of FOXY3. So to kind of start wrapping up, what is the clinical relevance of FOXY3? In order for us to further do more studies, we had to create a stable clone, a stable cell line. That means after 8 days, it doesn't go away, or FOXY3 expression does not reappear. So we have been working on two methods, CRISPR and shRNA, which is small hairpin RNA. And what these two methods done, I'll specifically focus on the lentivirus is what we used. When we do the transduction method, it's actually going to go in and incorporate itself into the genome, the DNA of the cells, and it's now forever there. Okay? And one of the indications that we use to determine, as you can see here, the top and the bottom shows you how the cells are decreasing over time. We treat it with the antibody that the cells that have the plasmid are resistant to. Hope that makes sense. So think about it like this. We add the vector to the cells, but all the cells are not going to take in the vector. So we have to isolate only the cells that have it. The way to do that is to treat it with the antibody, because the cells that ingest the vector have a gene that makes them resistant to it. So when we treat with pyrimycin, the only cells that will survive are the cells that took in our vector that will knock out FOXY3. And so we get it down to a single cell. We go from 1 million plus cells to a single cell. And then we have to expand that single cell to ensure that the population of cells we're looking at all have FOXY3 depletion. Once we do that, then we, of course, culture the cells, do RNA isolation and purification, reverse transcription, and then real-time PCR to detect the levels of FOXY3 and to confirm that it's been knocked out. So you'll see on the top and the bottom, I've labeled pre-shutdown, post-shutdown. This is more so to highlight that our method and our technique works, okay? And you'll see in the top, the black bar is just the normal PC3 cells that we've already established have high expression of FOXY3. The other two are two different clones that we were able to kind of drill down on. And the standard is you want to have at least 70% knockdown of expression. So this was promising. We had to repeat this again. And so in the bottom graph, you'll see that we drilled it down to about 7, I believe. The two that we are now focusing on in the lab currently are the two that I've put a box around. And actually, those two gave us an even greater decrease in expression. One was about 90%. I think it was clone 0.2. So we've been able to successfully knock down FOXY3 expression and establish stable clones for our study. So let's kind of summarize this and give it clinical relevance. This was our proposed model that we have established for our lab based off of some of the work I've presented today. And that is FOXY3 is an integral member of creating this prominent or rich soil in the bone that helps to allow these prostate cancer cells to thrive. We even believe that it's not just in the bone, but it actually can be in earlier non-metastatic cells. But we think that this is an interconnected relationship because various cells can express FOXY3 and it can be modified or modulated by being stimulated through other growth factors. So what our role is next, some of our future directions, and actually is current because we're currently doing this now, is we're identifying pathways that FOXY3 is involved in. So we're taking those clones and now we're doing strategic knockouts to see if we knock this gene out, what happens, so that we can create and identify a signaling pathway specific for FOXY3 expression. The other thing that we want to do is look at the clinical relevance of it by using an in vivo model. That means we're going to put it back into the mice. These are knockout cells. And look at how the cell tumor grows. Does it grow at all? And then lastly, we're working on a hemp project where we think that may be helpful because there have been studies that show hemp is anti-cancerous. So we want to actually look at what fragment of the molecule is actually stimulating this retard in cancer activity and how it will tie back potentially to FOXY3. So I want to say thank you to, these are some of my previous grad students and undergraduate students here. I didn't have enough room to put all of them up there, but I could not do this work without them. They're in the lab, they're working hard. Some of them have been here overnight. So I want to say thank you to them. I want to thank the Office of Sponsored Programs for this opportunity to present my research to you guys. The Macaulay and Roca Lab at Michigan, the Yates Lab at Hopkins, the boss, Sig, for supporting my research whenever I go to him. Hey, I need this. He's always been very supportive of getting the supplies I need. I want to thank Dean Taylor because alongside Sig, he was very instrumental in saying yes, or yes, you need to do this, no worries. If I'm having a problem, he was very supportive of that. So I want to acknowledge them as well. And of course, I could not do this without funding. So I want to thank the El Supardi Foundation for funding my research, the Faculty Development Fund, which I heavily relied on when I first came, and my grant from the Department of Defense for allowing me to do this work. Any questions?
We have pretty much in common, so I have a few questions. First of all, about the technical aspect. So you mentioned that you used, of course, lentiviral vectors to deliver small inhibitory RNAs, and you mentioned that it was complete knockout?
Not complete. You never get a 100% knockout, no.
Yes, of course, it was downregulation, right? And yes, and then I have another question. So I know that students are working with CRISPR-Cas in your lab and pretty much. And my first question is, are there any data about haploinsufficiency of FOX3 gene?
No. So not pertaining to cancer. No, there has not been any work on it. So anything pertaining to FOXY3 and cancer is going to come from our lab because it's not being studied.
All right, I understand. So then, are you using CRISPR-Cas to try to knock out it completely?
That model is being created now.
Probably I didn't see. It might be here data. So what is efficacy of CRISPR-Cas if it's not haploinsufficiency? So you know that it's and how you select after all cells as opposite, right? In case of small inhibitory RNA, you have the pyrimidine resistant gene, right? And it was kind of, well, the signal, so you could select. What about CRISPR-Cas? What is efficacy of, let's say, of knocking out this gene? You know, 2 alleles should be targeted, right? Both of them
Correct.
should be eliminated. That's what it is. So how would you select the cells? And what's the efficacy? How many cells in the population approximately?
So,
Activated?
okay, so that technique is being done now, so I can't answer the efficiency of it because we're still testing it. One of the issues with using CRISPR to knock out FOXY3 and what we've experienced is it is a small gene. So we are having issues with it being more efficient, and we're using another collaborator in Canada to help us to create the stable clone using CRISPR. So we're using 2 different methods to show the same thing. So unfortunately, I can't answer your question because we haven't finished doing studies on it.
And there are off-target effects. Oh, no, okay, all right, I'm finishing. Dr. Farangia, please.
Yeah.
Before you used CRISPR, didn't you do it restricting enzymes also?
Well, we used inhibitors to look at the behavior of it, but we weren't able to finish analyzing the data because, yeah. Yes.
I was just interested in knowing how you became interested in this line of research.
So I initially in grad school, we had to do rotations. And I said, let me work with microbiology. No offense to anyone in microbiology. It just wasn't my thing. Salmonella, I decided early on that's not for me. And in my first year in grad school, my grandfather passed away from prostate cancer. And I vowed to be an advocate for men because a lot of times there's so much research and so much financial support for breast cancer, but oftentimes prostate cancer goes unnoticed, unfunded, and I just wanted to be a champion for prostate cancer research, and I've been doing it ever since.
When it comes to the cannabis part of the future, is it kind of exploratory at this point, or do you have an idea of specific metabolites?
Yeah, it's still exploratory. We haven't even officially started because we have to go through the legal aspect of it. But just hypothetically, what we're trying to do is first establish and purify the molecule, and then I'll try to work with chemistry to identify the component of it. So it's going to be a very multidisciplinary project. Yes.
Speaking of multidisciplinary,
Yes.
you mentioned working with pathology to get the Gleason scores for the tissue.
Yeah.
What were some other professionals you worked with, and like what kind of roles did they play? I know you mentioned pathology. Did you work with ortho, or like what other areas of medicine and science?
Thank you for asking that question, and this should go for the students that are interested in medicine and you feel like I don't want to do research or I don't know what I want to do. Everyone has a hand in doing it in a collaborative manner. So in order for me to go into the clinic and harvest the bone tissue from the patients or to collect their blood samples, I work with a neurologist in the hospital. So even if you want to still see patients in practice, you can still collaborate with researchers to help them gain access to human specimens. Another, again, Dr. Grizzle, whom I'm very grateful for, trained me at UAB. And all we did for hours upon hours was look at tissue. But it helped me understand another level of the mechanisms of how it works and connecting it back to clinical relevance. So for you all that want to go into the health professions, you can still and always have a connection back to research. And I always tell my students the best physicians are going to be the physicians that are also scientists. Any other questions?
Jackie, I'm not an expert in this field, but is this the same FOXY3 that regulates hair follicle?
Yes, yes. So that component is great, and one of the reasons why we went to it, because that process is what we call embryogenesis and development. So this protein is not just responsible for bone regulation, but it's also responsible for turning on and off genes at early points versus late. So the easiest way to explain it is our cells have this genome of various genes that can promote proteins, but they're not always expressed at the same time. And when cancer cells become aggressive, they turn on genes that were initially off. and they utilize and they hijack those developmental factors to substantiate their growth. And so this is why my lab looks at it from an interdisciplinary approach. We look at wound healing, embryogenesis and development, bone remodeling, and the immune system, because they're all connected. Any other questions? Mario.
What do you look for like clinical relevance and like actually what do you want your lab to like produce that could be actually used?
So one of the things I had on the future direction slide, but I took off to reduce the amount of information, was a peptide. So what we want to do is work with Dr. Jesse James to create a peptide of FOXY3 and then treat the cells with this peptide and the mice to see if we inhibit FOXY3, what is the role? And that can potentially be the start for some type of therapeutic delivery. I want to say it's 119 amino acids. It's not a very large protein, and that sometimes makes it difficult. However, there are isoforms of it. So we try to focus on one specific, and as our lab grows, we'll move forward from it. Yeah.
Have you guys looked at the expression of the different isoforms in different types of tissue? Did you notice anything specific to bone?
We didn't, but one of the guys who's probably the father of FOXY3 in development, he sent me some vectors to look at the various portions of it. We just haven't gotten around to it yet.
I know you mentioned about men not going to the doctor. I know there's some, just in clinical practice, there's some indications that perhaps black men are having more aggressive forms.
Excellent.
Are you looking to that if there's expression?
Yes.
more in black men than
Actually, thank you, Dr. Carter, for asking that question. A portion of our research that we sent in for the grant is looking at health disparity, a health disparity component, which is looking at individuals that are at a greater risk, or number one, in mortality and morbidity, and that is African Americans. And so the issue is, because they don't often go to the doctors as much, getting those samples becomes very difficult. So the first thing that we're going to do, in addition to analyzing tissue specifically from African American men, is to do community engagement. So Leanne has been setting up a relationship with the School of Nursing so that we can go into the community and not just do bench research, but inform them of preventative ways to reduce cancer progression and to catch it early. So if there are any men watching or in the room, it's important to have your prostate gland checked annually, and also be very diligent about doing it, because it will be a matter of life and death.
That's kind of one of the my first year out in practice, one of my patients, when we diagnosed, he came in and he had bone mets.
Yeah, yeah. And that's one of the probably unfortunate things, is knowing that the men that you work with this are going to die. So the goal is to just increase their lifespan, even if it's 2 months, 3 months. But again, going to the doctor, getting checked up is really important. Any other questions? Well, thank you guys for listening and tuning in, and I hope you've learned something today. If you have any other questions, I'm happy to answer them afterwards, or you can shoot me an email. And again, thanks to OSP for this opportunity.
Trojans Talk Research is a monthly spotlight on the research interests of choice faculty, presented by Troy University's Office of Sponsored Programs.